So, okay, so today, as already discussed, I'll be talking about mass spectrometry and how that drives our glycoconjugate analysis and our investigation into structure-function relationships in infectious disease and also in vaccines. I just give a brief overview here. So between 2017 and 2023, we really looked at a range of different research topics, and they're kind of just outlined here, and I'll get to what we'll really focus on and some future endeavors in the next couple of slides. So we looked at, as far as in analytics and vaccine conjugates, we really looked into differentiation and quantitation of linkages in glycopeptides and free glycans. And these were mostly really revolved around salic acids and their particular linkages. And I won't get into that too much in this talk, but especially the 2,3 linked and the 2,6 linked salic acids, the differentiation between those two is really key in a lot of biological processes, including infectious diseases. We developed some glycan array technologies, and for us particularly, it was for C. elegans. And we've developed a range of informatics, and those include a glycopeptide analysis tool, which we termed GLIMPS, M-Salad, which was kind of a visualization program to help us with our array work, and then MS, excuse me, MS Assign, which is really a MALDI-TOF-based bioinformatics program. As far as glycosylation monitoring of seasonal influenza, we looked at some of the reference standards, which are really representative of the antigens that go into the seasonal vaccine. We looked at a range of vaccines as well, and to characterize glycosylation patterns on those. And then we looked at cell substrates. So when oftentimes vaccines, for instance, those that are subunit vaccines or those that are virus, killed virus, split virus, and so on, they're generated in cell lines. So you probably know, many of the people that are here probably know that the cell line used really dictates largely the glycosylation patterns and the types of glycans. So those were of interest to us, obviously for vaccine purposes, and also the basic science versions of those analyses. So we looked at MDCK cells, different versions, those that had differences in 2,6-salic acid. And then we looked a lot of CHO cells, the glycomics of CHO. So we worked with Dr. Bettenbaugh, Mike Bettenbaugh, who's done a lot of glycoengineering of CHO cells. And then we looked at structure-function relationships in viral immunity and vaccine development. We really focused a lot on coronavirus. We actually looked at coronavirus years before the pandemic, and we were looking at avian coronavirus, and then the outbreak came, and then we all kind of dove into that. I won't be really expanding on those topics today, but what I really want to focus on is the clumix analysis of a range of different avian and swine influenza and their glycosylation patterns, and what that really says or infers about an interaction with the innate immune system, specifically the lung surfactant SPD, which is a key first-line defense in respiratory tissues. So our current direction and focus, kind of one of the two major areas, excuse me, I'm getting over a little bit of a cold, is hemagglutinin glycosylation, that is an influenza, and what happens when these things arise in avian or swine and move towards humans. And so we've identified, we have identified through our informatics that we developed in-house, really a range of scripts and other bioinformatics programs. What interesting strains are available in the databases. where glycosylation sites may change, the receptor binding domain amino acids may change, and the regions whereby innate immune lectins and other lectins in the immune system may interact. So these are all different areas of these proteins, and the glycosylation on those regions really makes a difference. And of course, the RBD is susceptible to amino acid change, and that's very key in jumping from species to species and in that kind of mixing pot. So we will do glycoinformatics, glycoprotein analysis, and then we will also look at, and we have looked at, you'll see some of the data here today, immune system lectin binding affinities in some of those lectins. And I'll show you today is two different kind of formats to look at that. Receptor preferences, so glycan array analysis, with some collaborators that we have, and also co-alignment of glycosylation sites, the glycan present, the range and heterogeneity at each of those sites, and how they align with antigenic sites. So getting back to kind of the immunity and vaccine issues and how glycosylation may impact those events. We're also kind of taking a step in a new direction, and one that isn't very well explored at this point, and that's multiple attribute methods development for polysaccharide conjugate vaccines. These are different than what I've already discussed. Polysaccharide conjugate vaccines are basically, in general, they're polysaccharides isolated from the bacterial capsules. These are antigenic, and they become much more antigenic and long-lasting immunity when they're linked to a protein. So this hence the conjugate, polysaccharide conjugate. However, they can be very complex, and there's a range of kind of standard techniques that have been used for decades, but nobody's really been able to apply mass spectrometry to look at multiple attributes of these drug substances, drug products, and their intermediates in their production. So we've now begun to focus on that. We'll do bottom-up type proteomics approaches and top-down, and we'll get a little bit into why we're making that approach, and we'll compare those to some of the standard assays. Okay, so this group probably is already fairly familiar with glycosylation, but I want to relate some things about glycosylation that are important to this particular talk. So N-glycan synthesis, the types of N-glycans, and the consequences of pathogen recognition events. So we all kind of know, I don't know if you can see my arrow here, but you can imagine that the N-glycosylate starts with the transfer of the canonical Gluc3 man9 glucnac2 to protein. That's quickly trimmed back to man9 in the ER. And then it moves on through the Golgi, the late ER, and the Golgi stacks for processing. And in mammalian systems, that gives rise to high mannose glycans, hybrid glycans, and complex glycans. Excuse me. Drink quickly. So some of the factors that are impacted by glycosylation are DC sign, which has immune function, mannose receptor, which also has immune functions. One of the innate immune responders. Sorry. SPD, which recognizes mannan and other saccharides, polysaccharides, oligosaccharides, and pathogens. Langarin, which is a dermal located lectin that's also linked to a very specialized dendritic cell in the skin, and a target for development in vaccine design, and of interest and overall in innate immune systems. It can also impact the antigenic site. So you can imagine if a glycosylation site is lost at an antigenic site, the response will differ. If it is presented in a non-natural glycan in that position, that may also alter the antigenic site, and then maybe possibly give rise to an inappropriate antigen in a vaccine setting. Viral receptors. So, for instance, in influenza will have a receptor binding domain that tends to bind a range of different glycans, and specifically either alpha-2 or alpha-6 terminated glycans, and also viral enzymes. So, influenza neuraminidase is a key enzyme that helps the influenza release and also kind of get through that mucosal layer to actually get to the cell surfaces where it can infect. So these are kind of aspects about glycans and glycan interactions that we're concerned with. Okay, so a little bit about cell substrates. So the cell type and the tissue origin can dictate, at least in part, the type of glycan that's presented on the cell surface. So in terms of N glycans, um, and going from less complex to more complex, and looking at three different kind of versions, mammalian and avians, insect, and then plant, these can give rise to different glycans. So the less complex would be high mannose across these kind of subdivisions, but then moving into the ER and into the Golgi and how those enzymes are presented and the complexity of the glycan that's resulting at each individual glycosylation site can differ. So if the glycan, the high mannose glycan, is not completely processed or is not exposed to all the enzymes, you can have kind of a hybrid glycan, which is a mix between complex and high mannose. And then in some of the more complex processing, hence the name complex glycans, you can give rise to multi-antennary glycans with a range of different monosaccharide components. Insects are a little bit different. They tend to have highly fucosylated cores. The glycosylation processing leads kind of away from the high mannose and a lot of processing of the mannose residues. So this is kind of a mid-range processing in insects, and generally speaking. But the core can be significant because 1,3-fucosyl core fucosylation isn't really seen in mammalian systems. So that can actually be a little bit of a concern as far as allergenic response. And then further processing, you can almost have a mammalian-like limited complex glycan or hybrid. Plants, again, are a little bit different. They can have a fucosylyl core. They may have more interesting complex glycans. So these are kind of representative of different cell substrates and cell types. Getting on a little bit to mass spectrometry. So what I'm kind of displaying here is a very simplified flow. I'm not going to go deeply into this, but just to say that there are kind of three different prongs in mass spec approaches for glycomics and glycoproteomics analysis. And that really to analyze what is the glycan, what is what's the quantitation there as far as the overall glycosylation. What is the site-specific glycosylation show? What are the local heterogeneities? How they may impact the target lectins, implications for the RBD, and so on. And then occupancy. So not every N-glycosylation site is 100% occupied. So it will make a difference. What is the percentage of occupancy at that site? So those are kind of the three prongs. These are kind of ways to get there. So this is less complex, but you can kind of see the general paradigm. Now getting more complex, this is from a review that I wrote with one of the folks in my lab, Lisa Parsons. And this is a few years old, so it may be slightly outdated, but you can see that there's a lot of different types of mass spectrometry experiments to generate a range of different types of data, all of which may overlap but have slightly different fragmentation patterns that are generated from glycopeptides, glycans, that can help us to understand better what the structures are that we're analyzing. So I won't get deeply into this, but my lab is equipped for most of these things. Again, we're looking at three different kind of areas. One's the glycoside, the heterogeneities, the glycopeptide, essentially characterization. And then we can look at the deglycosylated glycans. And we can also look at essentially glycosylation site occupancy and so on. So this is just a basically kind of overall general diagram of what's kind of available for analysis. Okay, so now I'm going to get into one of our main stories, influenza, hemagglutinin glycosylation, and pattern susceptibility to lung surfactant SPD. So modeling and glycosylation sites. So going into the PBD database, there's a rich data data available there where you can look at the X-ray crystal and other forms of structural characterization that gives you the basically the molecular presentation of these antigens proteins. So here we're looking at kind of the glycosylation sites, how they're placed around this kind of stick and ball and ribbon representation. We can kind of investigate the receptor binding domain and other domains. And these information are used in combination with our structural data, our biological experimental data, and the reported properties of the investigated glycosylation and structure of these proteins in, for instance, recognition events. So this helps us to understand the protein and the glycosylation sites. So we looked at a range of avian and swine influenza, and I'll tell you a little bit more why. So going back some years, we had looked at a Mallard Denmark strain that was of pandemic concern, or there was concern that it could jump to humans. And when it had jumped to humans in Southern Asia and the poultry workers, it was very dangerous. I can't remember the exact morbidity, mortality, but it was fairly high. So we knew from our previous work and the literature that site 165 in the head of HA always, as far as what people have revealed, has a high mannose glycan. And this was related through Kevin Hartshorn and others to be a site where SPD interacts around 2000. Now, when we looked at the H5, what was, 165 in this particular instance and 162 in some others, it was always complex. So that would not be a ligand for SPD. So is this a target for SPD or not? And where does the difference exist across these different avian strains? Because this would obviously be a concern on a possible pathogenic marker. So looking at the crystals, this is a superposition of the H3 and the H5, and you can see that H5 has a lot of contacts with the 220 loop of HA. So it's really compact in close to the backbone and probably not very flexible, probably not a lot of motion there. Now, looking at the H5 and glycosylation site is slightly downstream towards the C terminus, and that puts it further out on a beta strand and not as interactive with that 220 loop. So we have very few contacts, which would imply that it's closer to the solvent and more flexible. Now getting to SPD, so what is binding to SPD? So this is some work by Crouch et al. I think Josiah was also involved in this from Boston University. And so it interacts with that high mannose glycan, and it really tends to be most high affinity for that MAN12 alpha 1,2 um, lower arm, which tends to form in this pocket, although other high mannose glycans are also interactive. And so I'm just kind of pointing out some calcium ion coordination between some of the residues, and then basically just pointing out where this actually binds. This kind of shows you the overall picture. Okay, so lung surfactant SPD in influenza. So SPD is a key immune colectin, as I've already stated, and it's present in lung and other respiratory tissues. It binds high mannose glycans. It serves as an important component of an initial barrier to infection. Hong Kong, 1968, was really where it was first really pitched out. And this was really in Ian Wilson's group, published by first author Skakel in 1984. So it's been known that that glycan is high mannose for quite some time, at least in those examined. So the 165 site's been identified as a key SPD interactor or ligand, and increases in the number of high mannose glycans at that head region really increases the interaction with SPD. So a little bit more about influenza. There are 18 H8 types and 11 neuraminidase types. It infects multiple species, including humans, swine, avians, horses, bats. And it forms kind of a mixing ball with these species. And through genetic shift resulting from reassortment of co-infecting viruses, new viruses can arise. So if you look here at the kind of virus genome, it's segmented. And if you have co-infection, you can wind up swapping some of these like chromosomes. So that can lead to a range of issues, and one such was in 1957 when a pandemic reassortment of the H2N2 and H1N1 gave rise to a new H2N2, and it was a pandemic, and over 1 million people died worldwide. So these events still happen, so they're always going to be a concern, and so the avians and the swine are monitored annually by different organizations. And so these are outside of the normal human pathogens, type 1 and type 3. And they can emerge eventually through these type of interactions to be potential pathogens. And so what are the susceptibilities to SPD? Because that's one of the first line defense, so we were interested in knowing this. So we looked at these different forms, H2, H5, H6, and H11. And particularly around this site, which is interactive with SPD. So this is the H2 through 11. This is an alignment. And you can see here the H3, which is kind of our model for interaction with SPD. The corollary and glycosylation site is slightly C-terminal. So it's a little bit shifted down. And then that 220 loop, we have different presentations. So in this case, and in other cases, so we have an arginine that's interactive, one of the key interactors with that glycosylation region and site, and also can also be a tryptophan. So what we're looking at here is that some shifts that infer a difference. Okay, so some of the mass spectrometry. So as I've already related, H3N2, we looked at that again, and really exclusively as far as we could detect, it was really high mannose at that site. H2 was exclusively complex as far as we could tell, and H5 similarly, H6 similarly, and H11N1, which there's not a whole lot of structural data, also was complex. So what does that mean for SPD? So what we partnered with Jeff Taubenberger's group at NIH, and they ran some SPD hemagglutinin inhibition assays, and this is just roughly how it works. So I won't spend a lot of time here, but you can imagine it works with red cells, and so you have your virus, and this is an inhibition assay, so it's incubated with various concentrations of SPD first, and then interacted with red cells. Red cells have the ligand on their surface, the glycan that the particular influenza likes to interact with. So if you have a good interaction with SPD, you get inhibition of agglutination. So these are the results from our study. We used two different HAs. I'm sorry, two different SPDs from the Hartshorn group, and then we also used two different types of red cells, human cells and turkey cells. And so, as far as the HAs here, here we have one of the avians, H2s, and then we have pintail, which has only that 165 glycosylation site. That is high mannose. And then we have a seasonal, which has more than one high mannose site around that region where SPD likes to interact. And then we have H5, H6, and H11. So you can see in this context, there was interaction with a lower amount of SPD compared to the avian strains. And similarly over here, even better interaction, and it really we really saw that kind of increased mannose have an impact. So it looked here as though these influenza strains, their HA, was really not interactive with SPD. So we've already kind of gone over this. We know that the H3 HA has a lot of interaction with that backbone, and likely results in poor processing in the ER and Golgi, since it's probably the glycan, the nascent glycan, is probably not interacted with those enzymes. And then here we have one example of H2, and there's fewer interactions. The glycosylation site is further down this beta strand, and there's fewer interactions with that 220 loop. So the hypothesis here that would explain the result is that that glycan is more mobile and more accessible to solvent and likely to the enzymes of processing. Similarly here, slightly different orientation, a few more interactions, but again, further down on that beta strand, probably more available, resulting in the complex glycan patterns. So in conclusion, the 165 is sufficient for SPD binding. The influenza recently appeared in human populations have this 165. H1 is very similar, has a very similar site in that head region. Avian influenza strains do have that complex glycan at 165. Therefore, they're missing the major glycan for SPD. So HA does resort. So knowing this probably gives us an awareness of how pathogenic these might be if they do tend to resort. And if you remember back to that 1957 version where we had H2, it's likely that had a complex glycan and may have contributed towards this pathogenicity. Speculating here. So absence of that 165 is likely a virulence factor. There's been a range of reports in the literature where if that site is missing from, for instance, H3, that strain tends to be much more pathogenic. H5 species, which lack that high mannose, have severe lung immunopathy. And so the data seems to align with the importance of that SPD protein. So we then looked at H4, which is another avian strain. Its fold is much more similar to H3. So we were wondering what's going on with this guy. And there was already precedent for a range of these strains that were circulating in Southeast Asia, where there was some mutations in that RBD domain that was moving it more towards swine and in some cases towards human receptor type. So the human receptor was primarily interested in binding to 6-salic acid, the preferred human receptor as far as we can tell. And then the dual receptor, which it would bind either 2,3 or 2,6, so it could kind of move back and forth between avian, swine, and human. And then the avian receptor, which was really preferred the 2,3 salic acid. So Okay, so it has some potential there if it does jump, and it seems to be liking the idea. What happens if it jumps? What happens with SPD? All right, so I'm kind of skipping a little bit closer to the punchline here. So this is the corollary to 165 in H3. This was really all high mannose. It was all high mannose. So at least these strains appear to present high mannose glycan. And so looking at the 3D structures, again, H3, we've already gone over that. The duct has more contacts. It's a little bit further up the beta strand, so it may have less contact with the solvent and then less probably less susceptibility to enzyme action. Similarly for this H4. So we looked at by SPR this time, we looked at interaction with SPD. So we linked the SPD to the matrix of the SPR platform, and then we analyzed. And what we found was that the binding KDs were really quite similar to Pintail and to the pandemic. So again, and then we actually knocked off a fair amount of the high mannose glycans with endo-F1, and then we saw a loss of affinity. So these are well correlated with our structural observations, and these tell us that H4 probably does interact with SPD. So probably less of a risk, but still. All right, so summarizing, H4 is making this shift, so it has this ability, and it apparently is mixing in that mixing bowl with avian and swine and human, either swine workers or other poultry workers, and it does seem to have this ability. Now, H4 infection in humans is rare, although we do see seroconversion. So there's not a lot of infection, but there is seroconversion. So it probably is making its way into humans, but it's not causing significant disease. These are surveilled. For instance, in Sichuan province, China, they found multiple co-infections. So it does seem to be mixing, as I've described. And viral HA contain this high mannose site, unlike the other avians that I've already described here. So has a different presentation. So probably is a substrate for, not substrate, but a ligand for SPD. And so we're predicting that it is susceptible to SPD. Maybe this or maybe it doesn't really explain, at least partially, why it is not a serious infection. So this first line, innate immune factors probably involved. All right, so I think I've already kind of gone over this, what we've kind of learned. I mean, I won't belabor this too much. All right, so excuse me. I want to get on to a little bit the future directions that my lab is taking. So we're going to continue looking at influenza. Excuse me for a second here. And we're going to kind of take several different approaches to really analyze this. And some of you already kind of seen some of the nascent approaches we have already done here. So we are in the process of identifying through house-derived informatics which IAV strains would be of interest. So which ones are really in this mixing bowl and showing signs of jumping to human. So we look at the RBD and the amino acid changes around that region, and we're in the process of picking representatives to then go forward and analyze. We're performing glycoproteomics informatics analysis for reasons I've already described. And we're also going to look at immune system lectin binding affinities. And we have partnered with Maurice's group, who's actually at this talk, and we're really looking forward to looking at some of these in our SPR setup with our collaborators here at FDA. And then receptor preferences. So we have collaborations with two different groups to do glycan array. And so we were hoping to see, differences in RBD, and then co-alignment with glycosylation and antigenic sites. So that's kind of along the lines of what I've already described to you. The other focus will be on this new multiple attribute methodology for polysaccharide conjugate vaccines. So I'll talk a little bit about these two. Okay, so again, we're looking to identify these epidemic and pandemic potential of some of these AIV. Again, we'll be looking at the shifts in glycosylation, the RBD mutation, lectin binding site domain changes, and then we'll choose representatives. And we partner with a company up here in Frederick, Maryland, to produce these. And we have our first samples on hand, and we're running some of those now. H8 proteomics, we'll look and co-align antigenic sites and influence on antigenic sites over time, and then determine the relative affinities of select to these various lectins. And hopefully that will help to inform us of pathogenic potential. And so we've coupled with these groups, as I've already mentioned. Maurice, that's attending here, is with Christoph Radinger at University of Vienna, and we just received some of those lectins recently. We'll begin optimization there. And then we'll select HAs by glycan array, and then look at those receptor binding site preferences. Okay, so here's an example of kind of a readout from an interpretation of our informatics approach. And so I won't get deeply into this, but what we've done is we've kind of segregated these into changes that are more preserved in humans compared to avian. So what we're trying to do is we're trying to find those where the amino acid changes in that receptor binding domain and glycosylation sites are appearing more and more in human. So it suggests that they're driving more towards that. So that's, for instance, one of the drivers in our selection. So we'll do that. We're looking at H7. We're actually currently looking at H5. Why H7? Because looking through the data and periodic changes, H7 does seem to appear, every 7 to 10 years. And last time it appeared was around 2015 to 2017. And it seems to be very busy in that mixing bowl right now. All right, so I won't belabor this too much, but this is kind of the approach. Database acquisition, Python informatics, to look at these 3 specific areas, choosing, and then off to production. And then we'll do these 3 sort of analytics. And here are essentially the deliverables, which you can imagine. We're looking to find out what are the patterns, how the glycosylation sites align with antigenic sites, what's the interaction with SPD, DC sign, and so on, and glycan array analysis to determine what are those patterns, and looking for intermediaries to that zoonotic leap. Okay, so moving on to the last subject, multiple attribute methods. Okay, so the overall structure of the resultant antigen varies in size. So we're making these complex molecules with a polysaccharide and a protein acceptor. Depending on the chemistry that's chosen, you can get very different structural products. The size of the saccharide that's linked to the protein varies across products. There can be repeats of 14 all the way up to hundreds of repeats. So the chemistry can really dictate the size and the complexity. So cross-linking and so on can occur in these things, making them more complex. So there are multiple conjugates per drug product. So we're up to 21 antigens per vaccine now. For instance, in pneumococcal vaccines, there's up to 21 and it's going up from there. So these are complex products. The structural data in the traditional techniques is really, it's always incomplete. There's a lot of background development work that goes into really defining these things. But if we had something that really digs deeper into the fine structure of the intermediates, the drug substances, and the drug products, this may better inform us, for instance, in differences not expected. For instance, presence of a ketone in type 14B in pneumococcal bacteria. And this modification can be from anywhere from 1 to 20% of the repeats. So you can imagine if that's part of the antigen, you're looking at large differences depending on the strain you choose. Okay, so a little bit about polysaccharide conjugates. So these are representative three polysaccharide conjugates. Here's an acceptor conjugating protein. CRIM 197 is his favorite. And then depending on the chemistry, you can wind up, for instance, if you have only reducing end linkage, you have a fairly simple conjugate structure. If you activate across for instance, the vicinal hydroxyl, so you have multiple sites down the polysaccharide, you can wind up with very complex shapes. And then again, even more complex shapes if you're actually linking together the protein as well. So, varying degrees of complexity, we're kind of taking a simple approach and looking at the less complex first. These are a list of analytics that are used traditionally. I won't get into these, but these are some of the comparators we'll use as we get into this project. So where does mass spectrometry really apply? So size and hydrodynamic volume, we can get information that is corollary to SEC mols. Composition and purity, that is applicable for mass spec. The extent of activation, that is the chemistry that activates the protein or the saccharide. We can definitely assay that with mass spectrometry, and so on. So mass spectrometry is really adaptable to this. While there is a number of hurdles that need to be overcome, and it will be challenging. But I think this will be well worth it to have the added advantage of that fine structural detail. So we will do. We've already generated multiple oligosaccharide conjugates to BSA. These were actually they're basically just 1,4,1,6 linked glucose. They'll be fairly simple. Then we have an MTA with a number of companies to look at actual products that are, or drug substances that are available in products, and where we have an understanding of the esters. And we'll do top down. So the intact conjugate, we will do some top down analysis to generate some of that data that we're after. And then frame 3, we'll do bottom up. So basically very similar to other proteomics approaches, we will digest these and then identify the site of identity, the site of conjugation, the saccharide heterogeneity site, and the percent occupancy. And for instance, in the intermediate stage, what sites in that protein, the target sites, are actually modified. So we can look at the step-by-step in the manufacturing process. Okay, so this is just kind of an outline of an approach. I won't get into this in too much detail, but we're taking a kind of an approach where we'll test a number of different orthogonal data to give us information in this process. The chromatography, we are in the process of trying different matrices to best suit the application. We're dealing with various chemical techniques to basically split apart the conjugate while preserving the protein backbone. That'll be enzymatic and also chemical. And then we'll also be experimenting with charge reduction, which can typically increase the coverage of these proteins by 10 to 15%. So these are kind of the approaches. Bottom up, as I've already said, we'll probably approach this with, again, with a range of different types of chromatography to optimize separations. And this dissociation strategies will at first include CID, HCD, ETD, and UVPD. These are different modes of fragmentation that really yields, different yields of fragmentation, different sets and subsets of daughter ions, and different mechanisms. For instance, in ETD, in um, uh, electron chain methodologies. You're basically generating a cascade where you're particularly targeting the peptide backbone. And UVPD is a higher energy application, which would be useful predictably in the top-down application. I won't get too far into this more, but we're kind of entering into this. We're hopeful that we can develop some techniques that will help in this subclass of products. So this is just the chemistry involved in our model conjugate. We have multi-oligosaccharides, which were isolated from a particular beer. We found a fortuitous presentation of 1,3 and 1,6 branches. The 1,6 makes it a little more soluble, so solubility won't be an issue compared to more of a linear 1,4 glucose polymer. And so we're using hydrazine activation chemistry. And so the sugar ring is open under basic conditions, and the hydrazide formation is followed by reduction. So we end up with something that looks like this, simple linkage, something that we should be able to generate marker ions for to follow. And so we're starting out with a very small, almost oligosaccharide conjugate, and then moving to larger ones that are 2 to 3 times the size. So we'll start with neutral polysaccharide, and then moving towards the more interesting charged forms. So this is what we're essentially hoping for. So we'll characterize several of these conjugates initially, and developing methods for neutral intact conjugate, free saccharide, and free protein. Adapt mass spectrometry methodology for charged intact conjugate, free saccharide, and protein. Develop mass spectrometry methods for proteolyzed neutral conjugate for site identity and conjugation site heterogeneity and occupancy. And develop mass spectrometry methods for proteolyzed charged conjugate for identity conjugate site saccharide heterogeneity and percent occupancy. And then data analysis to compare these to, for instance, standard techniques such as SEC mols, which really generates size of hydrodynamic volume for the protein that's not linked, the saccharide that's not linked, and then the actual conjugate, and then high field NMR to follow structure. So these are some of the preliminary data. These are the pools that we've then linked. This is some NMR experiments. We're seeing here is that this is the hex 14 standard. Has a little bit of 1,6 linkage. You can see here that there's much more linkage of 1,6 in the two model saccharides. These are now being linked to the protein. And this is a carbon protein HSQC, which allows us to help us to map the structure of these saccharides. And this is the ongoing work. So I basically already described this. We're working on the bottom-up approach first, and chemically similar compounds, analysis of intermediates, so that linkage region, digestion analysis of the glycopeptides for site occupancy, heterogeneity, and sequence coverage. So that's what we're about. I hope the presentation of our analysis of the influenza HAs was useful to demonstrate what we're trying to do here. And these are acknowledgements for the range of collaborators and folks that have worked with us on these projects. So these are the members of my lab, past and present. Structural biology. This is the NMR lab run by Daron Friedberg. And then my boss, Willie Van, who is in polysaccharides and who also contains Robert Lee, an expert on the conjugate chemistry. Ziping Yi in viral products. He's been very helpful as a co-author and collaborator in our in our influenza work. Jeff Tallenberger at NIH, Boston University, Kevin Hartshorn for his work in SPD in collaboration. Utrecht University, we had done a lot of work in coronavirus and some in influenza with these folks. And then our facility biotechnology core, those at Beth Israel, Rich Cummings, Jamie Heimberg, Molinaro, and then, of course, our new collaborators at Chem Proteins. So thanks, everyone, for your attention.